actually.
He's not staying afloat. That's the thing.
Have you seen his fucking GPA? He's not getting into med school, respectfully.
Okay, so a little later that I hope that we finally posted the human genetics assignment.
Let me see.
Bigger than it actually is. But what you're really just going to be doing is
Okay, ready?
using online sources to find out information about the human
So
genetic
basically,
trait that's caused by a single gene.
this is the general
And then
mechanism,
you're gonna
right?
find information
So
about
this
how
is
they
called
identified
like
that
6
gene
pi
before we're going to
because
we'll be talking about some of those methods
there's
today.
1,
And then also,
2, 3,
once they figure out
4,
what the gene is,
5,
how does the
6.
encoded
Okay?
protein fit with the disease? You know, linking
6
function
electrons.
to the disease.
Okay?
And
But
you know,
that doesn't
put on
matter.
there kind of
But the
additional way you
mechanism
can do it by going to online
basically
resources,
is
but
you
you can
have
also get
this
AI to help you with that as long as you say
plus
it actually gives a pretty good answer. Gives good answers. But the one thing to think about is that a lot of times when you work on this project, a lot of people like to pick a disease that maybe they read about in class, they have a specific interest in that disease, but not all diseases have a single gene responsible for it. So if you can't answer the questions easily, I advise actually picking a different disease trait. Some just get it complicated. Okay. Okay, so with that in mind, we're going to talk about kind of some classic genetics, molecular genetics, on how did you go about figuring that a particular gene in the DNA is what's responsible for it. And one of the first ways to do
So
this
in a sense,
Hey!
it is the general mechanism, okay?
Top
In
100?
a sense,
idea.
E
Okay.
plus
That's too
replaces
The idea is that if we can figure out what chromosome, what protein in the
Okay.
gene our disease is linked to, then we know that our disease gene must be in that region, and we can then see what predicted protein.
right?
So one of the first, I'm going to go through like a theoretical couple of slides first.
How can I talk
So,
to you about this?
you know, if you think
Okay.
about many human diseases,
Grandpa.
you may have heard that they are
So
higher frequency in certain
that's just
populations.
in general, okay?
And the reason
When
is
you're
you
like
might think it's
predicting a
associated with a particular
product.
population.
But
And the
the
reason for
actual
that is that you have
mechanism,
a geographically isolated population, and there's a new mutation that arises that results in disease. So that new disease allele is occurring in the genetic context of that population. So whatever alleles were already present in that population are now also in this new disease. The person now has this new disease.
right? And then you have your E plus
So that
right
means
here,
that
right?
remember last time we talked
That-
about, you know, these 3
This
million
double
variant
bond
sequences
is
you have
gonna
in your
come
genome
and
that are really,
attack
you know,
this,
not
the
necessarily
E
unique to you,
plus.
but are from your.
And then when
your
it does
pedigree.
that, this is called the
So, you know,
slow
we saw like Watson and
step.
Winter and
And
K
do you
plus
remember
White,
from
right? They each had their own
Chemical
set of
102? The
So
slow
if we
step
knew
is
which
what?
particular
The
allele
rate
was present in the
determining
population
step.
before the disease occurred, and then had individuals after the disease occurred, we can look at individuals in a family from that population who have the disease and don't, and look for genetic linkage to a specific SNP locus.
So
So
then,
what do I mean by that?
so when it
imagine
attacks
that,
that,
we have, in
it
this
will
example
look
here,
like this.
we have two different, and this doesn't have to
the
be humans, any
E, so
organism.
the double bond will
You have
disappear.
two genetic
And
backgrounds.
then
Blue
the
background,
E will
pink
attach
background, okay?
itself here. There's
And
an
what that
H
means
here.
is just that
There's
Well,
one
population,
H here.
different populations, they have their own set of alleles.
Remember, in
So
the
you can see that
beginning,
there's an SNP locus,
like
I call
here,
SNP1,
it was,
and in the blue background you have the A allele.
there's a one H here, one H here, right? There's a carbon and a carbon. And then there was a double bond, right?
In the blue background,
And then,
in the blue background you have the SNP1
and how many
allele.
bonds does
In
carbon
the pink
have in
background you
here?
have the SNP1B.
4.
Four, right? So there was 4 bonds for
maybe,
both
you
of
know,
the
T instead of
carbons.
C. Okay, that's
So
all
there
that
was a double bond
Now let's say
here,
the
there's a
mutation occurs in
hydrogen
the blue
here,
background.
there's a carbon here. That's 4
And we're going to say
bonds.
that M
So
is,
this here, there's a
you know,
double
set
bond, so
away
that's 2 bonds, there's a hydrogen, and then there's a carbon right here, right?
else back to it.
That's 4 bonds. But when you remove that, when you remove this, but then you add an E here, but you don't add anything here, what does this become?
Also give him a Asia, do you have a MacBook
So
charger?
let's say, so in other words, what happens is a new
My
mutation
MacBook's
occurs.
bad.
What type of
Why don't you
Mac
take
do
You
you,
don't know exactly
what type of Mac do you
That
need?
away from the SNP1 locus.
I
And
guess
because
so.
they occur in the blue background,
Hold on. I think I have a charger that could fit, but it's like really short.
So if we make an M1
Wait, let me see.
population
Asia, do you have one?
carries little m and
Asia,
heterozygous.
Do you have a
And then
MacBook
if
charger?
offspring derived from this, we may end up
You have a
with,
MacBook
two or
charger?
three
like a USB-C.
that become homozygous
Like, do you have the
for
plug
little
there?
m for disease. They'll also be
Is it in your
homozygous
bag?
for which allele problem, right? If little a is linked, if SNP1A is genetically linked to the new mutation,
Can I have something in here?
which allele will
Can you
usually be
plug it in?
inherited with little m?
Uh,
The A or the B allele? And if it's on set mutation,
Wait, can I have something in here though?
parental allele.
May I?
Those
Shown
snacks,
here with Rodney.
it's a salad, and those other 2 are for my sisters.
Here
Oh,
for
okay.
homozygous for
Fair
MS.
enough.
And what are they also homozygous for? Little a, SNP1A. Now, if this SNP was on a different
God
chromosome,
damn.
it would show independent
Huh?
assortment with disease. Okay? So what you want to think about here is
I
going
would
back to
throw
genetics.
it.
But instead of having 2 phenotypes, okay,
Can I have the
So
rest
when
of
you
the
remove
black
this
body
double
and
bond
more
here,
eyes and
what
front side, we're talking about an SNP allele
what do
and
you
a
put
disease.
right here?
They're still going to follow the rules of linkage.
Carbon, carbon.
So that's the principle of how many of the first human disease causing genes were found. You would have a pedigree with many individuals
There's a
who
carbon,
have
there's
this
a plus
disease
right there,
and many who
right?
don't. And then you'd screen individuals for co-inheritance of SNP's alleles all across the genome, okay? Many different chromosomes. So this next slide kind of shows you a summary of how that might go.
Right here,
So
a base
in humans, we're
is gonna
forgetting about
come
the
here,
sexual
okay?
trait for those who have
So
already
we have
probably
any
figured out
base
from the inheritance pattern. But an autosomal trait,
is gonna come attached to this
SNP
hydrogen,
markers on 22
and
and
then
23
this
chromosomes, 21 of the 22
hydrogen is gonna
chromosomes
shift
will show
and make
independence.
a double bond.
But what should happen is
Okay? And then
those on a chromosome that also
the
has
final
disease
part
will start to show
will
higher, will start to show genetic linkage. In other words, percent recombination getting lower. So what this example shows you here is a chromosome of about 200 centimorgans, right? Remember, centimorgan is percent
it's gonna be
recombination.
this.
And what was identified here were two SNP loci that showed about 20% recombination for the disease. That means they were co-inherited 80% of the time. And those, because those are SNP loci, they are actual pieces sequences of the DNA. That's the key point here, is that when you know
Now,
SNP
we're
allele,
gonna go to
you know what the
this
sequence
is
is.
called
When you have a disease,
hybridization.
you don't know what the
There's
sequence is yet.
different types of
So what they do in the slide here is they first find
The first
two
one we're gonna
SNPs
start with
that
is
are genetically linked.
chlorination.
And then they look at more SNPs, shown here with the little dotted line, the dashed line. And as you get genetically closer to your disease, you're going to see more and more parentals. So do you get to 100% parentals, no recombinant?
Oh.
Then this is where the big jump comes. Let's say that these two SNP loci here that I'm
So
circling
chlorination,
show zero recombination with the disease.
the same
You know
thing.
then that your
This
disease
is
gene has to be
the
in the
thing
area
is that all
where those
the
SNPs are
mechanisms
located.
are basically
And if
the
one
same.
SNP is on one side and one is on the other, you know that your disease gene has to be one of the genes located in between the two.
Right, and then you have
So when researchers are working on it, they call this like the zero recombination pit, okay? Because this can be millions of bases, right? But it still means it narrows where you have to look.
Boston
And what would
University.
happen is in humans or mice, our genome, you get down to this zero recombinant area, and you figure out what genes were present based on sequence. So remember we talked about, questions you would ask, is the gene transcribed? Does the predicted protein look like something else? Okay? Those are all good hints that you've actually found the gene. Once you found it, how do you know that that particular candidate is disease causing?
there's
And
before
we'll talk about some other
anything
diseases later,
happens,
but what they did here is
this has
they
to
took the
happen,
three
okay?
candidate genes
So you see the
and
CL2
sequenced them, comparing them to disease, people who have disease and those who don't. What would you expect? If you really had a
The
mutation in
CL,
one of them, that would probably be disease causing gene. Okay?
they have
So that's what they show you here. Okay, the difference correlates with the phenotype. I've taken that slide and just kind of annotated it a little bit, just to show you like that those markers here would mean like basically percent recombination. So what you see is that as the SNP marker gets closer to the gene, the percent recombination decreases.
not like
The
the
idea is
actual
that it would decrease all the way to
mechanism,
to
there's
zero.
just a preset
And then
that
that
you
pinpoints the
have.
region where the genes will be located.
After
So your
you
three
just,
candidates in this case,
it doesn't matter.
one of them
This
you have to make a case
but
for
just
is that
know
we're really
that
disease causing.
what you end up
And
with
it is kind of like making a case. You're
what you
trying to figure out what
actually
evidence supports one of those
is a
three.
CL plus
In this example, it happens to be
and
three
then
genes
you
because this is usually
have
what
a
happened with
CL
fibrosis. They narrowed it down to three different genes,
and
and then they had to figure out which one it was. Okay.
Okay?
For other disease genes, it might be
This is what you'll end up with.
not as obvious. Okay.
So all you have to
So,
know is just this thing.
So you've got these candidates. How do you narrow down which is the gene that actually carries the causative mutation of the disease?
Then now
Well,
we can do it.
you first try to predict what genes are
we
present
have this,
using things like BLAST. Things
our
like that.
original thing,
Um,
there's the H.
You'd look at the predicted proteins that are made and ask, do they encode some enzyme or activity that's consistent with
Is gonna
disease?
come
So,
and attack this.
in the case of cystic fibrosis, that wasn't necessarily obvious. They had three predicted proteins, but with cystic fibrosis, it's a buildup of mucus, and mucus membranes don't behave the way they normally do. You can imagine in a metabolic disorder where there's a specific enzyme, you might see that the predicted protein encodes an enzyme and things like that.
And you have
Um,
H here.
You look for the mutation, okay? And that sounds easier here than it actually is. Remember, there's a lot of variant sequences just present in the population anyway, and so you want to make sure you find the one that specifically would change a function to make it a mutant function. Okay, that could be a stop codon in the middle of a sequence protein.
Then there's when the CL.
Another important feature, okay? Remember we talked about how genes are transcribed in different tissues, right? Well, you might expect that a disease involved in having problems with mucous membrane ability to produce mucus would be involved in respiratory tissue. And sure enough, when they were narrowing down their choices for cystic fibrosis, one of those genes was expressed in lung and respiratory system, and say that became their prime candidate. The other thing that sometimes can happen, but not all the time, is that the mutation changes the size of the RNA or the RNA. And so we talked about what happens if you mutate the splice site, it might change the size of the RNA. Or if you have a mutation in the promoter, the transcription level might go down. And finally, this is something that might be done in tissue culture in a mouse model. You can, if you had tissue culture cells from an individual who has disease, and you could see symptoms of the disease in the tissue culture cell, you could try adding back in the wild type gene to see if
Same
that's
shit is happening.
functional.
Next one is bromination.
Not, you don't have to show all of these points, but these are just some of the questions you would ask to make the case that one of your several genes linked to disease is really the disease-causing gene. You want to think that there's only going to be one genetic cause, so only one of the candidates should actually be a disease-causing gene. So doing this type of
This
work,
makes sense, right? You see
diseases
how
like
it's
cystic fibrosis and also Huntington's disease, were, and also I think BRCA1 and breast cancer, were some of the first new disease genes identified or cloned by positional methods like this. So one of the things you may think, if you think about it, those are three diseases that are relatively, they're rare genetic disorders, but there's lots of individuals who have those genes. So that means there's a large population of people to test. Okay? And in fact, for cystic fibrosis, there's unfortunately an island off Venezuela where there's a closely related population where Huntington's disease is very prevalent. So this pedigree here, you can see, is kind of unique, right? But what that also means is it gives you a lot of, gives you a lot of individuals who have Huntington's disease, but it also means you have a lot of genetic samples that you can test. And when they found out that one particular SNP marker that was named G8 and one particular allele, allele C, okay, seemed to be present in all the individuals who had Huntington's disease, okay, in this pedigree. Couple of points, okay? Huntington's disease is dominant, so you only need one copy. So that's why you see a lot of times people who are affected have just one C. But the other thing you may notice is there are sometimes individuals who have the C allele who don't get Huntington's disease. So any thoughts on why that might be, that you have individuals who have the C allele but don't have Huntington's disease? What's something we talked about previously? Remember incomplete penetrance, okay, or variable expressivity, okay? It could be that Huntington's disease is displaying it in that individual. We know that there's an age correlation with Huntington's disease. But the other thing is kind of more basic. Remember I said that a new disease allele occurs in a population that already exists. So what's most likely here is that the first person who got Huntington's disease mutation happened to have the C allele of G8, okay? So that means that now the C allele of G8 is linked to the Huntington's disease mutation, but there are other people in the population who have allele C of G8 who don't have Huntington's disease. And so this individual could just be somebody who happens to have the C allele but wasn't related to the person who has Huntington's disease, okay? So the SNPs pre-exist in the population, all those alleles exist. The disease happens to appear in one person who has a particular allele makeup. So if for our purposes, we only always want to think of the disease, in these kind of rare, or not rare, but population-based studies, that one individual in the population has a disease. So you can imagine that there's lots of reasons why this method might not work. So that incomplete penetrance is one example. So that means that people you're testing, you think don't have the disease, but they do. So you're going to misgenotype, you're going to misassign people. Certain human diseases sometimes can be caused by environmental conditions. So you might think two individuals have the same disease, but one person has a genetic cause, the other is environmental. You can think of many types of cancers where there's an inherited version and also a new mutation occurring. Breast cancer is an example of that.
Can I lower the
Genetic
volume?
heterogeneity, that means that
For the
more
genetics
than one
lecture?
mutation can cause the disease, and so you might be looking at two pedigrees who have the same symptoms, but the genetic
No,
cause of one
Nick,
pedigree is different than the
your
genetic
side
cause of the
is
other pedigree.
good.
So when people were first working on BRCA1, they didn't know about BRCA2 yet, and so different families being studied ended up arriving at a different gene. And finally, a lot of diseases are more severe with other alleles from other genes, kind of epistatic effects, okay, enhancing or decreasing the effect of the gene. So these are just kind of points to just say like why this hasn't found every disease, but it's certainly been useful for many cases. Okay. We're now going to go through some modeling examples of what you would expect or not expect, okay, in inheriting, or what types of pedigrees are useful for mapping a disease allele. And some of this you'll see reminds us of when we were setting up test crosses and things like that to see if we could map recombination or not. So in this particular pedigree here, we're looking at an SNP, we're going to be looking at SNP1 linked to a dominant mutation called neurofibromatosis, okay? It's an autosomal dominant trait. And we're going to look at different pedigrees with different SNPs that are thought to be linked to the disease and see examples where we have useful information and then others where we don't have useful information. And what I mean by useful is that individuals who have the disease will have ambiguous genotypes with the SNP, which doesn't give you any sure linkage or not. So let's look at an example first where every individual is informative, as we say. So the cross in question is this one here. We see that the individual who has the disease is heterozygous for the GT allele of the SNP1. That's just the basis, G and T. Okay? And if we look, and then the other parent is homozygous for G and doesn't have the disease. Okay? So that means that every offspring from this cross is going to get one G from the mother, okay, and then a G or T, and neurofibromatosis or not from the father. And what you can see here is that most of the offspring who have neurofibromatosis, okay, 3 out of the 4, have another G. So that lets you model that probably the neurofibrosis mutation is on the same homolog with the G allele of SNP1. And then once in a while, you have a recombination between the mutant allele and G and T, and you end up with an individual who's like this, so they had a recombinant. Okay? But this is kind of a
Because they
small
said
version
majority
of
of
a
the
cross that
test
will
going
be
to be EAS
so you conclude from this that SNP1, the G allele, is on the same
Right.
homolog as the neurofibrosis mutation, and you'd expect most of the time to inherit the neurofibrosis mutation and the SNP1 G allele together. But you can see this is almost as a test cross, right? The father is a dihybrid for GT and neurofibrosis mutation or not. The mother is homozygous for the SNP allele and for the wild type neurofibromatosis. Okay? So this is
Yeah,
this
the
particular
final will
pedigree in SNP,
emphasize the new
everybody's
material,
informative
but
project.
will be cumulative. All previous, okay,
Now, in this example,
EAS
we're going to see,
reactions, halogenation, nitration,
Sometimes you get informative
sulfonation.
information. sometimes you
Yeah.
don't get
Okay. Activating and
And so-
deactivating groups in EAS reaction.
what you're seeing here is that
Renaissance.
if individuals who are homozygous, okay, for the SMP allele, okay,
New
they're
key
going to give you the form of
mechanisms.
information.
EAS.
Whereas if you are heterozygous for the SMP allele, they're not. And what's the reason for that? Well, you can get a AC combo from either parent, okay, right? So A and C, A can come maternally, A can come paternally. C can come maternally, C can come paternally. So these individuals don't help, okay? And this is what the cross would look like. So if the SMP genotype is shown on the left here, you do get information, but if the SMP progeny is on the right, you don't get information from those. In this example, okay, where the person with neurofibromatosis is homozygous for an SMP allele, you get zero, none of the progeny will be informative. Because remember, imagine we did a cross where you're big A, little a, an individual with big A, little b, little b. You can't tell what's recombinant and parental because the second gene is homozygous. So that's what will be happening, right? So no matter which SMP you got from the parent on the left, the father, it's always going to be A, okay? So you can't detect linkage. You can't show it to be recombinant. So the idea here would be that these would be 3 examples of SMPs linked to neurofibromatosis, but each one has a different usability in the particular parent. It all has to do with the genetic makeup of the parent. So another area, so in these examples so far, we're looking at pedigrees where we have a particular disease being inherited that we already have evidence is due to a single gene. So in other words, it shows up as autosomal recessive, autosomal dominant. But we also know that there are diseases where there could be more than one gene. Okay? But we can still use linkage to SMP alleles as a way of identifying parts of the chromosome. And this is something you might have heard about called genome-wide association studies. And it's based on the same principle that if you have a large population, in terms of thousands, tens of thousands of individuals, some have a particular disease phenotype, some don't. You might expect to see common parts of chromosomes linked to individuals who have disease.
These are descriptions of positions of an aromatic ring.
As opposed to the
All
rest of
the
the
reason this
chromosome.
ring is aromatic because it has 6 pi electrons. So this is the type of
So,
molecule that we're going to focus on
in
for
large
this aromatic
populations,
substitution.
thousands, millions of
There are three positions that are relative to this pre-existing
combinations
R group,
that
but
were
some types
established
of
early in evolution. This gets back to the idea that different
So the first ones that
populations
are
have
directly
different
next to the
allele combinations.
aromatic ring, this R group rather, are referred to as
When
the
you have a
ortho
block of
positions. These are ones
alleles
that
closely
are
linked,
directly
you inherit
on the
those
target, right next to that R
haploids.
group. Okay, right there.
So then you look at particular alleles,
Those are
haplotypes, that
so-called
co-occur
ortho
with disease
The
individuals.
next one we contend with
This
is
will
called
give you
meta.
linked parts of
This just means
chromosomes, but
that
it could be
relationship
biased just on the population.
this
But it does
group,
at least give you
a new
something
group,
to
and that
start
carbon,
looking
that's
for
relative
a
to
given.
the
So here's like an
pre-existing
example in
R
theory
group.
how this
And
might
the
work.
final
So
position that we
we're
have
looking here at
is
just
the
3
position
chromosomes,
called para,
and the red,
which
the blue, and the
means
green.
that it's kind
And
of directly
we have a
across
choice
the
of 2
aromatic ring
alleles
from the R group,
at,
right? Directly across from
or
that
10
R
SMPs,
group.
okay?
Right,
So the
so you see in
SMPs
this
are
particular
numbers
molecule,
10 and there's an A,
I've
B,
got
A,
two
B,
ortho,
each, keeping it
two
pretty
ortho constituents.
And so
two meta,
for individuals in the
and
population,
one para.
okay, who are unaffected,
Of course,
you'd
with the
expect to
original R
see,
group, that means that
occurrence
there's 6
of
total
both
positions
alleles
that you
behave
have,
with the
right? So 2 ortho,
allele.
2 para, 2 meta, and 1 para.
But if particular alleles
So
are
when we do
linked to
like co-aromatic substitution type reactions,
you might see
what we're basically talking about is if I have my pre-existing
statistical
group,
correlation
Y
between
in this
one
case,
particular SMP locus
and I do my
and a
electrical
particular
aromatic
allele. So in
substitution,
this
some
case
type of
here,
E+,
what we see is
I
that
could make,
for the individuals
in
affected in the
principle,
population,
okay, in principle,
while
I
they
can
show
make
both
3
alleles for most
different
SMPs,
Now,
we
which
see
one is this one?
enrichment for the 7B allele.
I
So
got
that might tell you that the
three
7B
descriptors.
area,
First one?
the SMP7 locus,
It's called ortho,
is
right?
nearby
Ortho,
where the
because
disease causing
the group
So this is sounding a little like
is introduced right next
calling,
to the
but
Y group,
what
right
might be
next to
happening
that
here
R
is
group.
in reality,
The
you
second
would
one
actually
is
get several different parts on the chromosome, which would give you
Refer back to
multiple
our
parts of the genome
picture.
locus. And also, it's
second
not
one
looking at a
is called
pedigree,
meta.
it's trying to use the whole population.
And my final one is called para, right? So it's ortho,
So-
meta, para in that order, right? Makes sense?
These approaches sometimes
So in
are like
principle,
called like a
those are
DNA
three possible
fragment,
products that one
trying to
can
find this one part of the chromosome that shows linkage.
Now, what we're
And
going to
so
learn about
when we look for linkage
in principle,
before, we
we
had
get some type of
we
focused
did
product,
a
depending,
square
it's
test
going to really greatly
to
depend
show
on what that Y
we
group
didn't,
is.
we
Right? So
basically tested
it's
to
not
show
going
that
to
we
give all of these
were not
products, right? So
unlinked,
good news
so we
is that
concluded we
we're
did.
going to try to come up with some
With
predictions
this type
for
of data,
what kind of
there's not
products
really
we'll
much of a
get.
chi-square test.
It's
You
not,
can
it will not
use
make
different
all
types
of
of
it.
methods.
But what we do is we
That's
sort of like
called
look at it in
the
terms of, first of all,
or
what are
log
these groups?
And
These were the,
what this is
these were
going
the
to tell
R
you
groups
is
or the Y groups that we
how
were
much
talking
more
about
likely
before.
is it that the
The
observed
green ball is
transmission
constituent.
pattern is due to
And what we're
the
going to do is try to
linked
identify or
compared to
classify
the
certain
bare
types of
bundle.
groups
So
that will direct the
kind of asking
reactions
the same question, but
into a
quite
certain place
differently.
in the aromatic ring. The first classification that we have are groups that are
And
so-called ortho
I'm not going to
para
ask you to
reactions. And
memorize
reviewing
the
real
formula,
quickly
but you want to know that
this
this
is the
name might show up in a
And
question.
then what we're going to see is that
this
ortho, there are certain substituents that I will talk about in a second that will
this
direct
formula
reactions
is the log,
give
okay,
both
of the
ortho
ratio. And
products
the ratio is the probability of the
and
observed
para
results
products.
occurring
So
at a
what we're
given
going to see,
combination
as you'll
frequency
see in a moment, is
divided
that
by the
ortho and para
probability of the observed
kind of go
results
together as a pair,
occurring.
right?
Okay.
It's kind of like
So we're still comparing
SN2
on
and E2 or something. It's like the two of those
So let's look
things,
at an
ortho and
example,
para, go
okay?
together as they're like a cousin, right? Does that make sense? They kind of like,
So
they get both.
we're going to
So
look
not
at
meta. So ortho and para are cousins, but meta is kind of like a cousin.
and
Now you might say, well, what are those ortho para directors?
Seven
The ortho para
individuals
directors
are
are technically groups that are very
so based with
electron
that
rich or
blended
electron
population,
donating.
we're
Right? So
going to
these
say that
groups
the recombination
are
frequency
all
is 12.5.
ortho para,
So this would be
para
an
directors,
SMP
and they're
allele
all
and a
some ways
particular
electron
gene.
donating.
So we've got 12.5% recombination. Okay.
So what are those?
So-
Electron donating groups are EDGs.
Now we're going to use this formula.
What are those? Right, so
We're
what
going
we've
to
got here
compare
are some
if
examples. We have electron
the
rich,
V's and
like
the
oxygens,
alleles,
like
the
oxygen is minus charge, or
are
OH
linked
groups,
based
or
at
OR groups.
12.5%.
These are very
Or
electron
consider
rich,
it
as well as the
what would happen
amines
is 50%.
shown here in blue, because they have they
So
have
the
these
probability of being
electron
unlinked,
both pairs on
would
the
be
heteroatoms. These
the
are
probability of being
hetero atoms.
R,
and they're all very strongly
which is
electron
a half,
dominated groups because they have those electrons on their heteroatoms.
to the X.
As well as
Okay,
these,
so in other words,
as these
any allele
isolated
that
versions,
you expect
this is an
to
ester.
half one, half
This
the other,
is
so
also
probability
pretty
for
strong,
any allele genotype is
electron
half.
dominated groups.
And you're asking that to occur for all
We have
else
some
equal,
other ones
so
just to
it
finish
has
off.
to
We
be.
have
So
alkyl groups,
there's
any
one
alkyl groups like
absolute
methyl or ethyl or
6
isopropyl
probability
and so forth
that you got
are
the
all
pattern
considered to
above
be
the
electron
allele,
dominated
right?
groups. Now they're not
If
as
they were
powerful
okay,
as
so in
these
other words,
here.
only people.
These have
So
electrons
if
that are
you
really
have 8
pushing as we'll
individuals,
see into the
7 are
heteroatom.
parental, 1 are recombinant.
And we also have sulfur.
If the disease and
And finally,
SSP
these halogens are
unlinked,
also, as we will talk about, are
P
considered
or
to
R
be
each have a proper
somewhat
probability
electron
of
withdrawing.
So
Somewhat
this
electron
pattern,
donating, I'm sorry.
7 P
They're
1
somewhat
R, would occur
electron
half
donating
to the 8.
in
Okay,
their
half
character.
for each possible. Think of it
So
as
these are very,
7
very
heads, 1
strong.
tail, right? It's
In terms of their
half to the
activation,
8.
these are the strongest.
Now, that's our
strongest on this side,
probability of unlinked. But what if they're
down to
linked at the
weakest over on this
12.5%?
side.
Well, if linked,
So
the
ortho
probability
paravectors,
of
that's
being
the
parental
concept we're pursuing
is
right now. As I had reviewed last time, I had mentioned to you
88
why is a group,
87.5%
like an
or
alkoxy
7/8,
group, like a
right?
methoxy group,
So
why
the
is that
7 Ps
a
occurred at
strong,
7/8,
what will happen
and the
right,
1 R
with
occurred
ortho
at
versus
So
meta
if you
versus para
multiply this
attack?
out,
And
it's
what we can do is we can do these attacks
20. So what did we
to
just
show
do?
all the
We took
examples
the
of an
same
attack that
results
occur.
of 7P, 1R,
The ortho attack
and
will
we
give
figured
us
out what's the probability of that
this is
occurring
ortho.
if they were unlinked,
I'm going
and
to
then we
place
figured
my
out the
plus on
probability if they were
that
linked
thing,
based on
and what we're going to
the
see
combination frequency.
is that that plus will wind up
So
being right
we
there
get
now.
1
Right
So now we put
underneath
that
this
into the formula,
one pair of electrons, and
divide
of course this is
120
very
by
stabilized. When this one pair is
456,
stabilized,
and we take the log of that value,
So the
it comes out
ortho,
as
the
1.
ortho,
So remember, we're talking
attack
about a
here
base 10
has the
log,
benefit
where each
of
value
having
is
this
10
very
times higher,
stable,
each log number. And so if we get a log score of 1.1, okay,
very strong or very highly stable, I should say,
that only 10, I
resonance
think,
structure
it's
that's
like almost
produced
a little over 10.
in reactions
Okay?
that work. That's
Usually
rationale
researchers
for why this
will
is
only
happening.
consider this significant if they get a log
Now,
score of 3, which
How
means
does
at
this,
least
how does this
1,000
relate to
times less
reactions
likely to be
that we
unlinked
do?
versus
So what we're going to do now is sort of see that when I do, for example, a nitration,
So
I'm going to
obviously
have,
in
for
this
example,
example I showed you
my
here, that would be
methoxybenzene, and I'm going to do a nitration
But the
using
reason
nitric
that
acid,
researchers
sulfuric
use
acid.
this score is because usually
And
you
what
are
we're going
dealing
to
with
get
small
are we're going to get
populations,
some type of
so your sample
mixture
size is pretty
of an
small.
ortho and para product. I would say that we tend to probably favor
So-
the para product a little bit more.
Also looking at
You'll get a
things
mixture of them,
as
you'll get the para product a little
theoretical
more, and this is
is
generally attributed to
we can kind of model
sort of like
out
steric
inheritance
effects, right?
patterns
The
of
para
what we
one
expect
is
to
a little
see
bit less steric
particular
inhibitor,
disease
but you're going to get both of those.
This is just to
Maybe,
remind us that
maybe slight,
cystic
alright,
fibrosis
slightly
is one
major
of the more
or a slightly
common
major product would be the para one.
genetic
But
cause
basically
disease
how this is
because
working
there
is
are
I'm getting
multiple alleles
the
across the
methoxy
population of the world. Okay?
is being
Unlike some of the
attacking
very rare
the
diseases where it
NO2
only occurred
plus
once, this occurred in
from
many
the ortho
different
and parasite. So the
So
ortho
it's
site
very likely
will
that
give us
if
this
you know
ortho
someone who
attack.
has cystic fibrosis, okay, they don't have the two alleles they have are not identical to each other. They're both in the same gene, but they're not identical to each other. That's sometimes referred to as a compound heterozygote. So anyway, let's look at this picture here of like what predicted patterns would be. Okay? And we're going to look through the left side first and the right side first. And the way they've color coded these is that gray are common alleles in the population. We might call those wild type alleles. And the colors are mutant alleles. Okay? And so if we're talking about a rare dominant trait, okay, not very common in the population, right? If we have two affected individuals in the same family, we probably expect that the disease allele would be the same in both individuals. But the non-disease allele could be two different wild types, because the maternal and paternal one could be different. So that's why you see two different shades of gray for their non-mutant allele, but the mutant allele is identical. Okay? If we look at unrelated individuals, okay, the two unrelated individuals may have different disease-causing mutations, but they'd be in the same gene. That's why they show them in different colors. And finally, the unaffected individuals, kind of our control, would always have different shades of the gray allele, the common wild type alleles. Okay? And this information is actually how, if you're trying to use genome sequence information to figure out what sequence is the cause of a new disease you're studying, you kind of might use this kind of logic. It's a little trickier when we're talking about rare recessive diseases.
And of course,
Remember,
this
two copies.
can have this very
So
strong,
within a
strong
family,
resonance
there's
structure.
probably going to be evidence of consanguinity. So the individuals who have the disease are going to be homozygous for the same mutant allele, two orange colors. Okay, remember, gray is a
Some
common
type
wild type
of
allele.
base, like
One of the
say,
common wild type alleles.
base,
Color is a mutant allele. Okay, if you have disease individuals, okay, from a family where there's no evidence of consanguinity, you might expect the allele, the mutant allele inherited from the father is a different base sequence allele than the one inherited from the mother. So they would be a compound heterozygous. So that's why you see all the children who have the disease always have an orange and a blue allele, meaning that one orange came from the father, blue came from the mother. Okay? So this would be something like cystic fibrosis, where many children who have cystic fibrosis aren't from consanguinity. Instead, it's just that the two parents each carried a different mutant allele. Okay? If you look at unrelated of individuals, again, cystic fibrosis might be an example, they will always have two mutant alleles, but they have collections of many different combinations of mutant alleles all in that same disease gene. And the control individuals are unaffected. They will be either homozygous for wild type alleles, the grays, or heterozygous carriers. Okay? So that gets us to a couple of points at the end of the chapter. You should read through these, and these are just, again, kind of based on the principles we've been talking about. I'll go through them here just to talk about kind of the logic. So what you want to see here is the same question is going to be asked a couple of different ways. A family comes in,
And then
there's two
similar
children,
is attacking
maybe two children have the same disease, but neither parent has a history of that disease. So you're trying to identify what's the genetic cause of it. And the fact that both siblings have it suggests that it's something that's inherited from one of the parents. Okay, or both. So here's an example of this kind of pedigree, right? So remember, with recessive diseases, right, it's very possible that neither one of these parents has someone in their family who ever showed this disease, but these two individuals must have been carriers, and they happen to have two children who are now homozygous for recessive mutation in this disease. And we're going to say it's what some research says. So can we use our modern technologies to try to help figure out where this disease gene, what gene is mutated.
So that's
Well, we can go with
how.
some of the logic we just said on
That's
the
how we're
previous
coupling
slide,
together, or we're
you would
associating
expect that
ortho
both of those
and parents,
children
kind of a little group,
have the
right?
same
They're both like
combination
cousins or
of these
cousin,
alleles
direct
compared
directions
to
for electrical aerodynamic substitution.
or any other symptom. And so, in this case here, the entire genome was sequenced from both parents and both children, right? And what was looked for, and I find this kind of a hard graph to read, but they're showing you two particular chromosomes, and it's kind of a summary of the data found. And so the light color across, okay, the entire chromosome across the like all red across, okay, indicates that both siblings inherited the same alleles from both parents. Okay, so that would be basically a 25% chance of happening. Okay? And so basically you can see here where it says identical. So you can see that this would eliminate you down to 1/4 of your genome, where both siblings inherited the same combination of alleles from both parents. Okay? So that would be a place that one could go start to look for particular diseases. Okay? And in this case, they were able to come up with candidates, and based on some of the phenotypes, okay, called Miller syndrome, okay, which I think had some kind of structural deformities, they were able to kind of make a good guess at what the disease-causing gene was. Okay, in another case, if you read the intro to the chapter, okay, they talk about a child named Nick Volker who has a particular disease, okay, and they wanted to try to do whole genome sequencing to try to figure out where the disease cause was. And so here's kind of, and this was all going to be based on sequence, no linkage.
So, and parents, kind of a little group,
This
right? They're
table
both like
presents
cousins or
kind of
cousin,
their logic
directions
of
for
how
electrical
they
aerodynamic substitution.
went from initially finding
You know, how is this
15,000
working in terms
variant
of like
sequences
energetics? I mean,
okay,
sometimes you see these,
that could be potential
maybe not your favorite sort of thing to be doing, but like
very
Brian's looking
interesting
at what we call reaction energy
So what did they do with
reaction
these?
energy diagram. What happens is that
Oh,
if I'm
and I
doing
should also mention that I believe in this case, they only looked at sequences found in protein coding. So in other words, that's why that number of 16,000 doesn't look like 3 million. 16,000 are what's found in only protein coding regions. Remember, protein coding is less than 2% of the entire genome. What was the logic for that? Well, most mutations have their effect on the gene by changing the way the protein moves. And so that was their logic. So they started with the 16,000. They looked for the change in the predicted protein, and if it changed the amino acid, they kept it. If it didn't, it was a silent mutation, they got rid of it. So that narrowed it down to 7,000. They then also looked at, well, across the whole population, how many of these sequence changes are found in other individuals with no disease? So they now narrowed it down to about 800, okay, that were not previously reported or new.
So that's what drives the selectivity. It has to be
They also knew this was X-linked, so they now look for X-linked and recessive, so they narrowed it down to 136. And then the last thing they did, the last couple of things they did, was they said, okay, well, if it does change an amino acid, does that amino acid change, is it something not seen before in other species, or is it just change it to an amino acid found in other species? The logic being that a lot of times, we can have subtle changes in our protein sequences between us and mouse and other primates, but those are usually called conserved mutations that are still functional, right? They look for changes that were never seen before in other species. So now they've narrowed it down a lot to 35, okay? And then finally look for very rare sequences of the mutation. And finally, they get to this mutation in a gene called X1AIP, which they think is the candidate, okay? And that's based on kind of what this protein is supposed to be doing, okay? Involved in, I think, some types of cell death and repair, okay? But that's where they're able to kind of find that. Both of these examples, you can see that there's a lot of logic used to go through the data, and also it requires a little bit of, luck is the wrong word, but fortuitousness, maybe it's luck. Anyway, I can't say that word. But good fortune in the sequence being in a gene that makes sense to the disease, okay? And so you can kind of read about this. And if you put this in a historical context, probably about 10 years ago, this type of approach of whole genome sequencing to find disease-causing gene, a rare disease, genetic, was starting to be done. And it was really done for as a proof of principle to show that you could actually do it, okay? It's still not the most common way to do it, because a lot of times you don't have evidence that the disease is necessarily present from one of the parents being passed on. It just could be a new mutation that occurs. So without some genetic data supporting it, you might not do this type of method. Okay? But so kind of in summary, just simply comparing your sequence to the published human genome sequence is still a very hard way to figure out what the mutation causing the disease is, just because there's already so much variation, okay, in the population. So you can see that you have to build a case of this. Now, why would you want this information? Well, once you figure out what the gene that's involved in the disease is, that might help you understand maybe potential treatments, okay, other things you can do to relieve symptoms and things like that. So if something is causing an anti-inflammatory response, maybe you can use anti-inflammatories, things like that. Okay. All right. So again, these last few topics I've gone through are more just to kind of get a flavor of the logic that's used to be able to go from whole sequence to identifying a gene. All right. So now we're going to have a pretty big shift in topics. Okay. So we've spent the last few weeks talking about both technologies and analysis of using genomes, predicting protein function, things like that. And we looked at whole genomes in kind of two ways. We looked at them in relation with inhumans or species to see how they could be related to disease or not, but also just in the terms of kind of evolution across species. Whole genome sequence is a way of studying conserved sequences and reconstructing whole chromosomes and things like that. Now we're going to talk more about kind of mechanisms where genetics has been used to help understand how the mechanism works. And so we're going to first talk about using genetics to understand how transcription is regulated. Okay? and this is a topic you've probably had the lac opera on before, but we are going to go over it again, but this time we're going to definitely approach it from the mutant perspective. Okay? And so we're going to not only learn how mutants were used in a particular way to study something, but also will give us a deeper understanding of how transcriptional regulation occurs. Okay, we're going to start with transcriptional regulation in prokaryotes because it's relatively simpler than in us and eukaryotes. Yes. Oh, thanks. I hope it just happened there. So a lot of points we're going to cover in this, okay? But we're going to learn a little bit about gene structure in a prokaryotic organism like E. coli. Okay? Remember, prokaryotes have no nucleus, okay? Usually single cell. Okay? We're going to see that genes are polycistronic, meaning that you can have one mRNA that encodes multiple proteins because you can have multiple Schindel-Darno sequences to initiate translation. Coming back to what we learned a few weeks ago. We're also going to see that regulation of transcription is not just based on the sigma factor and RNA polymerase, but there are other proteins that can help it do its job better or block it from happening. We're then going to see in a couple of the processes looked at is in bacteria, a lot of the genes that are regulated are parts of pathways that do common things. And if you're a single-cell organism, one of your main jobs is to get energy and produce nutrients, things like that. So we're going to be talking about regulation of catabolic pathways, okay, lactose utilization, and also talk about some biosynthetic pathways such as making the amino acid tryptophan. So it helps to kind of think of the ability to regulate a gene as almost kind of a simple circuit, okay, maybe on-off switch. Okay. So you remember from a few chapters ago, we learned about how RNA polymerase is brought to the promoter region using a protein called sigma factor. And we talked about the sigma factor recognizing a sequence at about -10 and -35. And we also saw that there was a consensus sequence, which was kind of the sequence that would occur the most often at a gene. Okay? And we hypothesized, and there's actually data that shows this, that -10 sequences or -35 sequences that are more divergent from the consensus don't work as well. Okay? So, but it turns out that what we can do is we can have additional proteins that can stop sigma factor from recognizing the 10 and -35 site, or help it bind it better, okay? And so these are going to be transcriptional regulators that basically help the initiation process happen better or worse. So first we want to maybe understand why we want to do this, okay? So we're going to look at a couple of different types of metabolic pathways. So we're going to have what we call inducible systems. And the principle here is that for enzymes involved in catabolism of a substrate, let's say a sugar, okay, you might only want to make those enzymes when the sugar is available, right? Because microorganisms especially, one of the key selection factors for species is ability to grow fast. Okay? And you don't want to waste energy translating proteins you don't need. Proteins are enzymes, right? So if you're going to make the enzyme for metabolizing lactose, which is a disaccharide, right, you only want to do that when lactose sugar is available. If there's no lactose sugar available, you want to keep it off. You don't want to use the energy. So sugarless utilization is a common example of what's called an inducible system. Okay? Substrate becomes available, you make the enzymes to digest. Okay, we also have repressible systems. These usually have to do with biosynthetic pathways, or sometimes called anabolic pathways. Okay? The production of nucleotides and amino acids, okay, are common types of biosynthetic pathways. Okay? And you may know this from our biochemistry, but bacteria need to make all their amino acids. Okay? In fact, they even do a better job than we do. We have to get some from our environment. Okay? But bacteria and yeast can make all their amino acids, so they have an enzyme pathway for each amino acid, and they make their own nucleotides. Okay? Nonetheless, I'm going to say they're smart enough. I don't really mean nobody needs smart, but they've evolved to be able to know that if there's free amino acids in the media or the environment, why bother trying to make yourself, right? Again, you want to save energy. So if something's available for you, okay, you turn off those pathways. There's thousands of lab experiments where people show that if you grow E. coli in media where you give it amino acids, it grows faster than what's called a synthetic medium where it has to make all of it itself. Okay? And again, it's energy conservation. So repressible systems, the enzymes for the biosynthesis of the metabolite, are feedback inhibited by the metabolite. So in the example we'll look at next week is that the presence of tryptophan in the media turns off or represses the tryptophan biosynthetic pathway. And then as you starve for tryptophan, you turn up that pathway to make it. Now, those are kind of two general models for regulation, or the whys of why you want to regulate. How you actually turn genes on or off, we're going to think of this as two kinds of circuits. One is called positive control. Okay? Positive, think adding something. Right? These are transcription factors that are called activators typically, and their job is to help RNA polymerase initiate transcription better. Okay? So they increase transcription. Okay? So if you're using a transcriptional activator, okay, it will show that activity when you need to turn on it. The other type of regulator we have is negative control. These are a group of transcription factors sometimes called repressors, and their job is to stop transcription from happening. Okay? Most genes are going to be regulated both by an activator and a repressor at different times possible. Okay? And there's different mechanisms for repression and activation. in the example of the lac operon we're going to talk about in a few minutes, okay, we're going to see that it has both positive and negative control. Okay, positive and negative control can be used for both inducible and repressible systems. Okay, so this is just kind of the way the switch works. Think of it, you have a brake and an accelerator. Okay, don't get one of the things that sometimes confuses students, and just make a note for yourself, is that the protein that stops transcription is called a repressor. Okay, repressible system is talking about the metabolic pathway where the end product can turn it off. Okay, so a repressible system can be regulated with a repressor or an activator. Okay, so the root of the word is the same and means turning off, but it's in different contexts. So let's kind of look at kind of the historically first regulated enzyme system that people study. And this is our lactose utilization in E. coli. We're going to call it the lac operon. And one of the reasons this was probably one of the first ones studied is that E. coli don't have to use lactose. You can give them other sugars. So you can have mutants that can't do this, or mutants that do it really well, okay? But it doesn't, you can still grow the E. coli. The other thing is that we have available chemical analogs of the substrate lactose or other regulators that either are toxic analog versions, okay, or can change color when cleaved. And so there's a lot of ways for helping to screen and select the mutants in the pathway. We're not going to go through those, but it's just very easy to isolate mutants.
Just touch
Now,
me.
what are the enzymes involved in lactose utilization? We're going to be focusing on two. Okay, the lactose has to get into E. coli. And so many organisms have either transport proteins or what's called a permease, which makes a channel, that allow the lactose to get in. So if you don't put lactose permease into your into the E. coli membrane, okay, you can't get lactose in. Okay, so that's going to be one of the required proteins we need to make the permease. The other protein we need to make is the enzyme that breaks down the lactose into two monosaccharides, galactose and glucose, which then feed into regular metabolism. Okay? So that enzyme is called beta-galactosidase. So we'll be talking about the production of both beta-galactosidase and permease as being necessary to be able to utilize lactose. So when researchers were studying this, they were able to assay these activities, okay, before they figured out what the genes were. And what did they figure out? Okay, well, there's the wild-type regulation of metabolism of lactose is that when there's no lactose around, that's the off state. So there's no production of the permease and galactosidase. We also have kind of our on state where we give the cells lactose, and they say, okay, let's get it into us. So they have the permease gets expressed, put it in the membrane, and then we can metabolize it with the beta-galactosidase. Now, at this point in time, going back, I guess, to the early 60s, researchers were mostly thinking that everything was regulated through enzyme activities. Okay? People weren't making the connection that you could actually regulate production of a protein by regulating the amount of transcription. Okay? Jacob and Monod, this is a picture of Monod. I just happen to like this picture because he's like just happy looking all the time. Anyway, but he was also very like politically active in things. But he decided to do a mutant screen, okay, to look for mutants that could not metabolize lactose, okay, or produce lactose, would try to metabolize lactose even when it wasn't around. Okay? So again, if we go back to our initial condition, the off and the on state, you can imagine two classes of mutants: ones that are off all the time and ones that are on all the time. Okay? And so you might imagine that mutants that were off all the time affected production of the enzyme. Right? Just the enzyme itself, and he found a lot of those. Right? The interesting one comes with the ones that are on all the time, constitutive mutants. Okay? And so this class here, okay, was kind of the surprise mutant, if you will, and the one that is going to lead to a model where it's actually regulation of transcription, okay, that is causing this to occur. And if you just think about this, again, what's happening here? Our normal state is off without lactose, on with lactose. If you have a mutant that has it on all the time, the implication is that, well, there must the normal function in the wild type, there must be something that keeps it off when in the absence of lactose. Okay, so that, and then when you add lactose, you remove that block. Okay, in these mutants, you somehow damage that blocking mechanism, so it's on all the time. Okay, so a general theme we're going to see is that proteins that keep a pathway off, okay, which we call repressors many times, okay, mutants in those will be on under all conditions. Okay, right? Think about it. If you broke the brake on your car, you couldn't stop, right? So think of an analogy like that. And so when they started classifying the mutants, they identified that one of these constitutive, on all the time mutants, was a particular protein that transcriptionally repressed the lac operon. Okay? And by knocking out this repressor, mutating it, now transcription was on all the time. So we'll kind of finish, I think, with this figure then. Okay? So we're going to show you a few different pictures here. Okay? The upper left is kind of the genetic structure that we're going to start with. Okay? So let's focus on that. Okay? The gene has a promoter region, okay, where RNA polymerase will bind. It then has an operator, which is the regulatory region where we're going to help or stop polymerase from binding. And then we're going to have the area that encodes three enzymes: LAC-Z, LAC-Y, and LAC-A. Okay? So digression for a second. Remember I said that in prokaryotes, it's common to have a single transcript made, an RNA made from one promoter, but then have that one RNA have the ability to code multiple proteins, because you just have to have 3 Scheindelgarno sequences to make 3 separate proteins. And so the 3 proteins made from the lac operon are LAC-Z, which is going to encode our beta-galactosidase, LAC-A, which is going to encode, LAC-Y, which is going to encode the permease, and LAC-A, which is an enzyme of really unknown function, but it's still made. And then we're going to be talking about a second protein called LAC repressor, which is coded in a different part of the genome from a gene called LAC-I. So what happens in the two different conditions? Remember, we had the condition when lactose was absent, and we have the condition when lactose is present. So let's look at the red box. That's the repression state when there's no lactose. So when there's no lactose present, this protein made from the LAC-I gene called repressor binds to the operator. It physically binds the DNA at a specific sequence, and it blocks RNA polymerase from coming to the promoter. Okay? It really is kind of a roadblock. Okay? So that's your off state. Okay? So in the absence of lactose, that's what it looks like. What happens when you add lactose? Well, you add lactose, the repressor protein is bound by lactose that you added, and it changes the structure so it comes off the DNA. That's a common theme in protein regulation called an allosteric change. The added chemical binds to the protein target and changes the structure and changes the activity. So in the absence of lactose, the lactose repressor is bound. When you add lactose, lactose binds to the repressor and it comes off the operator, allowing transcription to occur. Now, and then after transcription occurs, you get production of more nucleotidesidase, the permease, and the LAC-A, which is the transacetylase. Okay? Now, this does imply that there always has to be a small basal level of permease, because otherwise, how would the lactose even get in the cell to make this happen? But, so starting with this, how did Jacob Monod ever figure out how to put this together? Well, they looked at their mutants, and this class of mutants that they got were what they call constitutive ones. These were the ones that were on all the time. Okay? And they found two classes of constitutive mutants. Mutations that were that actually knocked out the transcription repressor itself, so it couldn't get made. And then they found ones that mutated the operator, which were called OC mutants. So here's how these mutants look. Okay? Here's our operon, okay? And here's a mutation that mutates the repressor, so it can no longer bind the operator. So this is called a LAC-I minus mutant. Okay? The other way we can damage this interaction is by mutating the sequence in the operator. Remember, the operator is a DNA sequence that the repressor recognizes. You change the basis so the repressor can't recognize it, it can't bind, it's on all the time. So we'll stop with this, but you want to come up with one fundamental point here, is that when a transcription factor binds DNA, it relies on the transcription factor recognizing the DNA, and the DNA being recognizable by the protein. So damage the repressor, so it changes the structure, it can't bind. Damage the DNA sequence, so the transcription factor can't find it, it won't bind. Okay? And then when we pick up in next class, we'll look at this to see what the structure of the actual LAC repressor looks like. Okay?
Slowly. It's done. Yours is done.